Understanding what goes into a lysis buffer is the first step toward optimizing your protein purification process and ensuring success in your downstream applications. Well-chosen buffer not only helps release proteins from cells, but also maintains their stability and functionality throughout the experiment. A typical lysis buffer contains several key components, each serving a specific purpose. Starting with number 1, components that maintain pH. Buffers like Tris or HEPAS maintain a stable pH, which is critical for keeping proteins functional and ensuring proper binding during purification. Number 2, you've got salts for ionic balance and stability. Salts such as sodium chloride or potassium chloride help maintain ionic balance, which optimizes protein solubility and stability during the process. Third, you've got detergents. These are essential for breaking up cell membranes and solubilizing target proteins. The type of detergent you choose can have a significant impact on your results. Fourth, you've got protein stabilizers or osmolytes. Purification processes can be harsh on proteins, so stabilizers like glycerol are often added to promote stability and proper folding. Fifth, you've got chelating agents. Chelators like EDTA bind to divalent cations, removing them from the solution to inhibit metalloproteases and prevent degradation of your target protein. Your buffer will also contain protease inhibitors. A cocktail of protease inhibitors is added to prevent degradation of your target protein, increasing the yield and purity of your final product. You would also have phosphatase inhibitors. If your protein of interest is phosphorylated, phosphatase inhibitors are added to preserve its native state throughout the process. Two main factors should guide your selection of the appropriate lysis buffer. These are target protein properties. Consider whether your protein is cytoplasmic or membrane-bound. This will heavily influence the type of detergent you should use. Second, you've got your downstream applications. Think about what you plan to do with your protein after purification. This will determine the specific components and concentrations needed in your buffer. The choice of detergent is often the component most influenced by these two factors. Choosing the right detergent can be the difference between a successful experiment and a failed one. Detergents in lysis buffers serve to disrupt the cell membrane, allowing the extraction of your protein of interest. Depending on the subcellular localization of your protein, you may need to choose between a mild detergent and a harsher one. For instance, proteins embedded in the cell membrane or located in membrane-bound organelles, such as the nucleus or mitochondria, are generally more challenging to purify compared to cytoplasmic proteins. When selecting a detergent, both its concentration and its type, whether ionic or non-ionic, must be considered. Non-ionic detergents are milder compared to ionic detergents. They have limited ability to disrupt protein-protein interaction and are thus considered non-denaturing. This makes them ideal for applications where preserving the native structure of the protein is essential, such as in protein activity assays or protein-protein interaction studies. Let's compare ionic versus non-ionic detergents. As non-denaturing agents, non-ionic detergents like Triton X100 or NP40 are commonly used to gently solubilize proteins without compromising their native folding or activity. These detergents are preferred when your protein structure or function need to be preserved for downstream applications like enzymatic assays. On the other hand, ionic detergents like SDS are much harsher as they disrupt not only the cell membrane but also protein structure by breaking protein-protein interactions. SDS, for example, is used in applications like SDS Page, where complete denaturation of the protein is required. Another thing to consider is the concentration of the detergent. The concentration of detergent also plays a critical role in solubilizing proteins while preventing aggregation and denaturation. Too high a concentration can result in protein denaturation or inactivation, while too low a concentration may be insufficient to fully solubilize the protein of interest. I would say these are the most important aspects to consider when choosing the right lysis buffer. Always have a look at what's inside your lysis buffer, have a good understanding of the protein you are trying to purify, and always consider what is the downstream application and if any of the components may not be suitable for that application. I hope this breakdown helps you choose the right protein lysis buffer for your experiments. If you found this video helpful, please don't forget to like, share, and subscribe for more content like this.